Students, we know that stomata are present on leaves and on other young parts of a plant. They are microscopic pores that are mainly concerned with exchange of gases and transpiration process in plants. Air enters the plant through these openings. Inside the plant body, carbon dioxide of the air gets used up for food production during the process of photosynthesis. Oxygen produced during photosynthesis is released through these stomata. Water vapors also pass into the atmosphere through these pores. Loss of water vapors from the aerial parts of a plant is known as transpiration. In dicotyledonous plants, leaves usually have more stomata on the lower epidermis than on the upper epidermis. As these leaves are held almost horizontally, the sun rays fall directly on the upper epidermis. Therefore, the presence of lesser number of stomata on the upper epidermis prevents excessive loss of water. The situation in monocotyledons is rather different, as their leaves are held somewhat vertically. The leaves of monocotyledonous plants have almost the same number of stomata on both their surfaces. Corn, wheat, grass are monocotyledonous plants having almost equal number of stomata on the upper and lower surfaces. Apart from the number of stomata being the reason for rapid transpiration, there can be some disadvantages of presence of stomata on the upper surface. These stomata may make the plant susceptible to entry of disease-causing microbes. Stomata on the upper epidermis may also get clogged by dust and mineral nutrients. Stomata on the upper surface may also cause mineral nutrients to be washed out of the leaf during heavy rains, and they may get clogged more easily during a dust storm. In our experiment today, we will study the distribution of stomata on the upper and lower surface of a leaf of a dicotyledonous plant.
Objective of this experiment is to study the distribution of stomata on upper and lower epidermis of a leaf. In this experiment, we will prepare a stained mound of leaf epidermis to observe the stomata present on both the surfaces of a leaf, that is, upper as well as lower. Materials required. Materials required for this experiment are a leaf of petunia, glycerine, safranine, forceps, petri dish, watch glass, beaker, brush, needles, blotting paper, blade, cover slip, etc. Procedure. Let us start with taking out a thin peel from the surface of the leaf. Take a fresh and healthy leaf from a petunia plant. Identify the upper and lower surfaces of this leaf. These surfaces are quite distinct. This is the upper side and this is the lower side. First, remove a thin peel from the lower surface of the leaf, as it bears a higher number of stomata. The peel can be taken out by tearing the leaf obliquely with a single jerk or scraping it with a blade. Drop these pieces into a watch glass containing water. Select a fine piece and place it in this watch glass. Let's wait for some time till the peels take up the stain. Take a clean slide. Carefully shift a stained peel onto this slide. Avoid curling of the peel. Gently spread it on the slide. Add a drop of glycerine over the peel. Cover it with a cover slip. Remove extra fluid, that is, stain and water, with the help of a blotting paper. Prepare 2 slides simultaneously. One with a peel from the upper epidermis and another with a peel from the lower epidermis of the leaf. In order to observe the prepared slides under a microscope, place a slide on the stage of a microscope. Revolve the nosepiece of the microscope so as to adjust the objective lens over the stage aperture. Locate the portion of the peel of the leaf that we are trying to observe and focus. We must first observe under low power lens and subsequently with high power lens. This is the slide of the upper epidermis, means a peel taken from the upper surface of the leaf. It contains lesser number of stomata. This is the slide of lower epidermis, means this is a peel taken from the lower surface of the leaf. It contains a number of stomata.
So what did we observe? In the leaf of Petunia, the number of stomata is more on lower surface as compared to upper surface. Or we may also say that stomata are present mainly on lower surface of leaves. We also observe that epidermis consists of a single layer of cells. Stomata remain embedded in these epidermal cells. Each stomatal opening, or we may say stoma, is guarded by 2 guard cells. Guard cells contain chloroplasts and a nucleus. We also observed that chloroplasts are absent in the other cells around the guard cells. While performing an experiment, certain precautions should be taken. Let us know the important things to be kept in mind while performing this experiment.
Precautions. The peel should be handled gently. Curling should be avoided as much as possible. The student should make sure that there are no air bubbles trapped between the slide and the cover slip. Excess of glycerine should be removed by using blotting paper.
Hope I have been successful in my attempt to demonstrate the experiment. Let us check through some questions what you have learned from this experiment. Question. From which surface of a dicot plant leaf would you remove a peel to observe stomata? Answer. In a dicot plant, stomata are mainly present on the lower surface of the leaves. Next question. Why do you think that leaves of dicotyledonous plant have larger number of stomata on their lower surface as compared to their upper surface? The answer is, dicotyledons usually have more stomata on the lower epidermis than the upper epidermis. As these leaves are held almost horizontally, their upper epidermis comes directly in contact with sunlight. Presence of fewer stomata on their upper epidermis helps them in preventing excessive loss of water. My next question is, what is the main function of stomata? Answer. You know, stomata allow the exchange of gases, carbon dioxide and oxygen during photosynthesis and respiration between the internal tissues of the plant and the outside atmosphere. They are also the main channels through which water is lost from the plant during transpiration. They can be closed to conserve water. Hope I have successfully demonstrated the experiment on making of a temporary slide of an epidermal peel of leaf of a dicot plant. Let us now concentrate on the second experiment to demonstrate the rate of transpiration from the upper and lower surface of a leaf. Transpiration is the loss of water from leaves and other aerial parts of plants into the atmosphere. It is a very useful phenomenon for a plant as it enables the plant to absorb more water through its roots. Transpiration pull is also developed because of this loss of water through leaves. The amount of water lost through transpiration varies greatly. There are a number of factors that determine the rate of transpiration. Transpiration rates go up as the temperature goes up. Humidity is another factor affecting transpiration. As you know, it is easier for water to evaporate into drier air than into air saturated with water vapors. Wind is also a factor which affects the rate of transpiration. Increased movement of the air around a plant will result in a higher transpiration rate. Most important factor is the type of plant. Different plants have different rates of transpiration. Some plants which grow in arid regions, for example, cacti and succulents, conserve water by transpiring less water than other plants. In some plants, the rate of transpiration is different from upper and lower surface of a leaf. Do you know the reason? More stomata are present on the lower surface than on the upper surface, especially in a dicot plant. Let us do an experiment to compare the rate of transpiration from the upper and lower surface of a leaf in a dicot plant.
Objective of this experiment is to compare the rate of transpiration from the upper and lower surfaces of a leaf using cobalt chloride paper. Materials required. Materials required for this experiment are a potted dicot plant, 3% cobalt chloride solution, a desiccator, 2 slides, 2 clips, filter paper, watch glass, etc. Procedure. To prepare cobalt chloride paper, take 3% cobalt chloride solution in a watch glass. Cut a filter paper into strips. Dip these strips into 3% cobalt chloride solution taken in the watch glass. Do it with the help of forceps. You see the paper strips turn pink. Let the paper dry. It is pink in color when wet, but turns blue on drying. Store the dried cobalt chloride paper in a desiccator to keep them dry. Or we may say that the desiccator keeps these strips free from moisture. Take out the strips from the desiccator only when the experiment setup is ready. Means when we are ready to use the cobalt chloride paper. In a potted plant, select a healthy leaf. Place 1 dried strip of cobalt chloride paper on the upper surface of the leaf and another strip on the lower surface of the leaf. Press them close to both the surfaces by using glass slides over both the cobalt chloride papers. Clip the glass slides to make sure that the strips do not move from their place. Keep the setup undisturbed for a few hours. Remove the clips, slides, and observe the paper strips. Compare the change in color of 2 paper strips. One from lower surface of the leaf and the other from upper surface of the leaf. Observation. During this time, there is some change in color of cobalt chloride paper strips placed on the upper and lower surface of the leaf. The strip on the upper surface shows little change in color, whereas the strip present on lower surface changes its color quite fast. Here we see the difference in color of both the paper strips. So, what do we conclude from it?
The quick change in color of cobalt chloride paper on the lower surface indicates higher rate of transpiration from lower surface as compared to the upper surface. This difference in the rate of transpiration is due to the difference in number of stomata present on both the surfaces. The lower surface has more number of stomata as compared to upper surface of the leaf. This variation in number of stomata makes the observable difference in the rate of transpiration. Precautions. Always select a healthy plant for this type of experiment. Store the dried cobalt chloride paper strips in a desiccator. Also use dry hands to handle these strips. Keep the setup undisturbed for sufficient time so that the results can be observed clearly. Hope procedure for this experiment is clear to you. Experimentally, you could prove it that the rate of transpiration is different on both the surfaces, and this difference is due to more stomata present on the lower surface of the leaf. Let us check what you have learnt through this experiment. This question answer session will also help you to face viva confidently during your practical exams. Question: how is transpiration beneficial to plants? Answer: perhaps the greatest benefit of transpiration to a plant is that it drives passive movement of water and minerals from soil to leaves. Transpiration provides the upward flow of water with nutrients. That way, the leaves get what they need. Question: how is transpiration different from evaporation? Answer: water is removed from the surface of the earth to the atmosphere by two distinct mechanisms: evaporation and transpiration. Evaporation can be defined as a natural process in which water is transformed into a gaseous state. This process requires large amounts of energy. On the other hand, transpiration is a process of water loss from aerial parts of plants through stomata. In most plants, transpiration is a passive process largely controlled by the humidity of the atmosphere and the moisture content of the soil. The next question is, what are the factors responsible for transpiration? Answer: transpiration is the loss of water from leaves of plants. Water leaves the leaf through stomata. Number of stomata on surface of a leaf affect the transpiration rate. The rate of transpiration depends on air temperature and solar radiation. Environmental factors like humidity, air temperature, wind, etc. also affect transpiration. Next question: is there any disadvantage of transpiration to a plant? Answer: too much transpiration causes wilting of the plants. Plants must have the ability to prevent too much water loss through transpiration. Usually, a waxy cuticle present on the upper surface prevents loss of water from this surface in desert plants. Secondly, guard cells have own mechanism to check the water loss. When water enters the guard cells, the cells become swollen or turgid, thus causing opening of the stoma. When the guard cells lose water, they shrink and this causes the stoma to close. Once a stomata is closed, there is no more loss of water from this closed stomata. Therefore, as such, nature has its own way to check excess water loss from plants, but if it is done, the plant will wilt. Hope you have learned the aspects related to stomata and the major benefits of transpiration. These demonstrations will help you to carry out these experiments in the laboratory more efficiently. Let us revise the procedure we followed in both the experiments. Distribution of stomata on the upper and lower surface of a leaf. Rate of transpiration from the upper and lower surface of a leaf. I hope both these experiments as well as theory behind them is clear to all of you. See you in the next practical class. Till then, goodbye.